So I would now like to welcome Stephanie Longet, who will be giving a talk titled Antibody Responses to mRNA Vaccine in Previously SARS-CoV-2 Infected and Infection-Naive UK Healthcare Workers Using Short and Long Dosing Intervals. Stephanie, if you would like to put your slides up and unmute yourself, you can go ahead.
So thank you for this presentation. I just need to make sure I share the right thing. Sorry, can you see my screen?
Right now we see it in presenter view, but if you go to display settings, you should be able to change that.
Yeah, I can move. Sorry. Yeah, the problem I cannot access.
Display settings is just to the right of that, at the middle at the top.
Yeah, the problem I cannot access. I cannot remove this bar. To see.
If you try to click the one that says display settings.
Yeah. Oh, yeah. Okay. Now. Yeah. I think.
Perfect. Please proceed.
Sorry. Yes. Okay. So thanks for the presentation. The introduction, I mean. So yeah, I'm Stephanie Longet. I'm a postdoctoral researcher in immunology at the University of Oxford, UK. And today I'm going to speak about the impact of short or long dosing intervals on antibody responses to Pfizer-BioNTech COVID-19 vaccine. So, as you know, SARS-CoV-2 emerged in December 2019 in China. It was a pandemic. And COVID-19 mitigation measures were implemented to reduce the spread of the virus and the impact of infections on population. The quick development of vaccines was key in the control of the pandemic. And the vaccines were developed using various platforms. So now we have mRNA vaccine, viral vector-based vaccine, and inactivated vaccines. All around the world, some research teams analyze the protective immune responses to COVID-19, including the PITCH consortium. So the goal of the PITCH consortium was to analyze protective antibody and cellular immune responses to COVID-19 following infection or following vaccination in UK healthcare workers. The PITCH consortium performed a prospective longitudinal cohort study in 5 sites: Oxford, Liverpool, Sheffield, Birmingham, and Newcastle. And there are now more than 2,000 healthcare workers who were recruited to date in the UK. The goal of the PITCH consortium was to answer several key questions, including this one. Does previous infection impact on response to vaccination? For the following questions, they are related to some decision of the UK government. Maybe some of you remember this. In December 2020, the UK government decided to delay giving second doses in order to give more first doses to more people. But at the time, we didn't know the impact of this modification of dosing intervals on vaccine-induced immune responses. So the key questions were, what immunity do people have during an extended interval between doses? And does the immune response differ between long and short dosing intervals? So as you know, both T cells and antibodies work in combination to control viral infections. So following an infection, we have first the innate immunity and also the adaptive immunity, including T cell responses and antibody responses. And today I'm going to focus on antibody responses, more particularly neutralizing antibodies. Okay, so to analyze the antibody responses and more specifically the antibody binding, we use a multiplex ELISA, a multiplex indirect ELISA, and we analyze the levels of antigen-binding IgG antibodies, and we were able to analyze the antibodies specific to 9 antigens, so SARS-CoV-2 spike, RBD, and nucleocapsid, but also the antibodies specific to other human coronaviruses, including the seasonal ones. We also analyzed the antibody neutralizing responses. For this, we use an ACE2 inhibition assay. It's a surrogate neutralization assay, also developed by Mesoscale Discovery. And basically, the principle is the same as the ELISA. But here, you have some variant spike proteins in the wells. And basically, it's a competition assay. So we add label ACE2, and we add our samples with the antibodies, and both can compete to bind the variant spike proteins. And we also analyze the neutralizing antibody responses using a live virus neutralization assay, and we quantify the reduction of the infection rate of the cell. Okay, so the first goal of these studies, because it's a combination of studies actually, so the first goal of these studies was to analyze the impact of a SARS-CoV-2 pre-infection on the immune responses induced by 1 dose of Pfizer-BioNTech COVID-19 vaccine. So we analyzed the immune responses 28 days following the first dose in UK healthcare workers, and we compare the immune responses in SARS-CoV-2 pre-infected UK healthcare workers or naive healthcare workers. Here you can see SARS-CoV-2 spike-specific IgG levels. In black, you have the immune responses in naive participants, and in red, in SARS-CoV-2 pre-infected participants. Open circle show IgG level before vaccination, so it's a baseline, and shaded circle show IgG level following 1 vaccine dose. And you have also another group in blue. They are naive participants who received 2 doses. And you can see there are significantly higher spike-specific IgG level in plasma of SARS-CoV-2 pre-infected individuals compared to naive participants following 1 vaccine dose. And these IgG levels are also higher compared to naive participants who received 2 doses. We also observed that 1 dose of COVID-19 vaccines induce cross-reactive IgG responses to SARS-CoV-1 and MERS spike antigens, and also to the human seasonal beta coronaviruses HKU1 and OC43. And we observe a particular cross-reactivity, especially in SARS-CoV-2 pre-infected individuals. The second goal of these studies was to analyze the impact of dosing intervals. So for this, the healthcare workers receive a second dose, either between 2 and 5 weeks following the first dose, or between 6 and 14 weeks following the first dose. And we analyze the immune responses at various time points following the second dose, and also during the long interval. And again, it's a comparison SARS-CoV-2 pre-infected versus naive participants. Here you can see SARS-CoV-2 spike-specific IgG levels in participants who receive the doses with a long interval. So on the left, they are the naive participants, and on the right, the pre-infected participants. And here you have the time points. So basically here you can see there is a decline of IgG levels during the long interval in both groups, but particularly in naive participants. But the second dose boosts SARS-CoV-2 IgG levels in both groups. We also analyze the neutralizing antibody titers, again, the Victoria strain, so the classical strain, and some variant of concern, beta, gamma, and delta. And here you have the results in naive participants who received the doses with a long interval. And again, similarly to the IgG levels, you can see there is a decline of neutralizing antibody titer during the long interval. And following the second dose, the second dose boosts actually the neutralizing antibody titers against Victoria, but also the variant of concern. And now it's a comparison short versus long dosing interval. So here you can see SARS-CoV-2 spike-specific IgG levels 28 days following the second dose. So again, you have the naive participants on the left and the pre-infected participants on the right. And here you can see there are significantly higher spike-specific IgG levels in participants who received the doses with a long dosing interval compared to participants who received the doses with a short interval. And this significant difference is only observed in naive participants and not in pre-infected participants. Similar results for the neutralizing antibody titer. Here you can see there are significantly higher neutralizing antibody titers in participants who received the doses with a long interval compared to these ones who received the doses with a short interval. And this is observed in naive participants and not in previously infected participants. And as you can imagine, there is a follow-up of this study. So we analyzed IgG levels 3 months and 6 months following the second dose. And now we are starting analyzing the IgG levels following the third dose as well. So what we can see on this graph, so again, on the left, you have the naive participants and on the right, the pre-infected participants. So we can see there is a decline of IgG levels over time, I would say as expected. But this decline is more important in naive participants compared to pre-infected participants. And following the third dose, you can see there is a boost of IgG levels in both groups. And finally, we are currently also comparing SARS-CoV-2 specific IgG levels in individuals who received the AstraZeneca vaccines. And basically what we observe that the AstraZeneca vaccine induced lower IgG levels compared to the Pfizer vaccine. And it's the case for all groups, I mean naive and pre-infected participants. But following the third dose, which is the Pfizer vaccine, because in the UK, the third dose is only an mRNA vaccine. Following the third dose in both groups, in all groups, we see an increase, a boost of SARS-CoV-2 IgG level. So now I would like to summarize the main findings of these studies. So first, we observed there were higher neutralizing IgG titers in SARS-CoV-2 pre-infected individuals compared to naive participants following a first vaccine dose. We also observed there was a decline of neutralizing IgG titer during a long dosing interval, and particularly in naive participants. But the neutralizing IgG titers are boosted following the second dose in both groups, but particularly also in naive participants. And finally, the most interesting finding, I would say, we found there were higher neutralizing IgG titers with a long dosing interval compared to a standard short interval in naive participants following the second dose. And this difference is not observed in SARS-CoV-2 pre-infected participants. So to conclude, pre-infection, the dose regimen, but also the type of vaccine, all these parameters impact on antibody responses post-vaccination. And you can find all results I presented today, but even much more. You can also find the T-cell data, which are very interesting. You can find this in 2 papers we published last year in Lancet Microbe and Cell. And finally, I would like to thank the PITCH Consortium and all people involved in these studies, and particularly Susanna, Paul, and Tushan, who led these studies, and my supervisor, Miles Carroll. And thank you for your attention, and thank you for giving me this opportunity to show this data, and I'm happy to answer your questions.
Thank you so much for that amazing talk, Stephanie. We do have some questions, so how about I have Dr. Fish start us off with a question?
Yeah, that was a great talk, Stephanie. Thank you very much. So some of this I can't remember from the papers, but I suspect it was in there. So in terms of the waning of the immediate antibody response, long versus short intervals, is it comparable that the response wanes at the same rate?
Basically, yeah, we see a waning in both cases, but maybe I can come back to the slide. Basically, we keep this difference. It's still significant. The difference observed between long and short is still significant. There is a little bit lower, slower waning with a long dosing interval. It's still significant. What is the impact, the clinical impact, I would say? We don't exactly know, but it looks, the waning is a little bit less important with the long dosing interval, but it's just significant. So.
Okay, and the other question, just a quick question I wanted, was the T-cell memory response. Do you see a difference in the extent of it in the long versus short?
We see a difference, and it's very interesting because it's the opposite compared to the antibody responses. We see a lower T-cell responses with a long dosing interval.
Okay, but they're still quite robust, correct?
Yeah, it's more robust compared to antibody, where we clearly see a waning. It's more robust. But it's interesting to see, we see the opposite, something.
And the memory response, do you know what that looks like?
So, yeah, for the comparison long and short, the work is in progress to see if we see some difference.
Thanks very much. Thank you, Stephanie. We do have more questions from the chat, from the Q&A section, sorry. The first question that we have is, have you noticed a difference in IgG titer levels in male compared to female and in different ethnic backgrounds?
Yeah, basically, we didn't stratify our cohort between male and female. And here in these studies particularly, we have like a bias because they are healthcare workers and they are more female. But it was not the goal of the study to compare male and female, but there is somebody in the group who tried to just to have a quick look, I would say, and there was no particular difference. And ethnic background here, I would say the ethnic background, there is also a bias. There are healthcare workers in the UK, so more Caucasian people, and so we haven't analyzed this in this study, so I cannot say there is a difference.
Okay, thank you. And you did touch on third doses in your presentation, but having that longer period of time between receiving a third dose and even a fourth dose, one of the questions that we do have is what might be the predicted effect of fourth dosage given a longer period of intervals and even immunocompromised individuals who may require further vaccines?
Yeah, the impact of the fourth dose. Now we are analyzing after the third dose. We don't know for the fourth dose yet. It's difficult to say. I would say it can be beneficial for sure for some immunosuppressed people. It can be beneficial, except for this one who cannot generate, for example, some lymphoma patients. It cannot generate any responses. So even if we give 4, 5, 6 doses, maybe it won't impact a lot. But for I would say most of them, it can help to boost a little bit more. My consideration, I would say, it's more between some people who can experience breakthrough infections, and if we add a first dose, it can boost a little bit more. But we are not too sure. It's unclear the beneficial role of the fourth dose. third dose, for sure, it impacts on neutralizing antibody responses, efficient against variant of concern. For sure, the third dose, it's clear, but more, we don't know exactly for the fourth dose. It's difficult to predict, honestly.
Well, thank you very much for all of your comments, for the questions, and for your amazing presentation.
Thank you.